Hi everyone. In this video, we're going to look at some of the techniques for cloning genes and how we can analyze those clones with PCR and electrophoresis. One of the most frequently used processes in biotechnology is that of making recombinant DNA molecules. We saw recombinant DNA previously when considering crossing over in meiosis. And in this process, we're going to perform a similar but more directed process to produce a DNA molecule carrying a gene from another source. To do this, we'll start by removing DNA from a cell and identifying our gene of interest. We'll then take a bacterium carrying a plasmid, a small circular piece of DNA which replicates independently from the main chromosome. Plasmids are relatively easy to isolate from the cell. And once that's been done, we can cut it and our gene of interest with restriction enzymes, which we'll look at more in a moment. Insertion of the gene into the plasmid produces a recombinant plasmid, which can be reinserted into the bacterium. Since bacteria have such a short doubling time, even growth overnight will produce a large population of recombinant bacteria, all carrying copies of our gene. Let's zoom into the actual cutting process for a moment. The enzymes that we use to cut the DNA are termed restriction endonucleases, or simply restriction enzymes. They recognize specific DNA sequences and cut between the bases in a set manner. In this example, the target sequence is GAA-TTC, which, as you can see, is a palindromic sequence. The antiparallel nature of DNA, along with complementary base pairing, means that the sequence is the same on both strands. Now, the most useful restriction enzymes for gene cloning are those that cut in a staggered manner, such as this one that cuts between the G and the A, producing a short, single-stranded overhang at the end of the molecule, which we refer to as a sticky end. These are useful for gene cloning because complementary base pairing specifies exactly how to match these sticky ends, enabling us to insert a piece of DNA with a complementary sticky end into the plasmid. Insertion into bacteria isn't the only way to produce a lot of copies of a DNA sequence. Polymerase chain reaction, or PCR, is a technique that allows us to copy a DNA fragment many times by virtue of a cycle of heating and cooling. In the first step, the DNA is heated to denature the strands by effectively melting the hydrogen bonds holding the bases together. The reaction is then cooled slightly to allow the binding or annealing of primers to the DNA. One way in which we can manipulate this process somewhat is to use primers containing the sequence for a restriction enzyme, so that once we've made all of the copies, we can insert them into plasmids or do other things with them. The third step is extension, which relies on a heat-stable DNA polymerase called Taq, which won't be denatured by the high temperature of the heating step. Taq uses the 3' hydroxyl of the primer and fills in the rest of the sequence using complementary base pairing, resulting in 2 identical DNA strands from the original parent strand. These steps are repeated as many as 30 times, resulting in around 1 billion copies of our target DNA sequence in a matter of hours. Now, there are a couple of problems associated with cloning genomic DNA, particularly when putting it into bacteria. The main issue is that of introns, which eukaryotic genes have, but prokaryotic ones don't. And so a bacterium won't be able to splice the mRNA to produce the necessary continuous coding sequence. Luckily, we have a way around this by starting with the completed mRNA instead of the genomic DNA. Obviously, that would leave us with the issue of having RNA instead of DNA. And so, we can use reverse transcriptase, found naturally in retroviruses, to produce a DNA copy of our RNA template, which we refer to as complementary or cDNA. Once the first strand is produced, the mRNA can be degraded, and DNA polymerase can synthesize the second, leaving us with a piece of double-stranded DNA containing only the exons of the gene, which can then be amplified by PCR and inserted into plasmids for cloning in bacteria. So after all of this, how do we check our cloned DNA? One way would be to use gel electrophoresis to separate out the DNA pieces to verify that it's the right size. In this procedure, DNA is loaded into wells in a gel, and an electrical current is used to separate the DNA. Since DNA is negatively charged due to all of the phosphate groups in the backbone, it will travel towards the positive electrode, termed the anode, with the smaller pieces traveling further through the gel due to less resistance. This can be used to analyze DNA for a large number of purposes, from verification of cloning to forensic analysis. Thanks for watching, and please be sure to check out the other videos in this section.